So the major focus of my research lab is to understand the structure and function of this major signaling pathway in the body called the TGF-beta family, or transforming growth factor beta. It's a major signaling pathway in all humans, and it does a lot of different things and different biological processes, all the way from development to homeostasis in the adult, and regulates numerous processes like regeneration, reproduction, wound healing, immune surveillance. I was actually the first ones to solve some of these structures of some of the ligands in complex with their receptors. And that's where I really got interested in this family of molecules, which is a very complex, very diverse signaling pathway. It incorporates over 33 different individual ligands. And so really the focus of the lab has been to try and understand how these 33 different ligands interact with their signaling receptors and how they're regulated by extracellular protein modulators. And it really has become a very important therapeutic target for numerous diseases. One of the major breakthroughs that we've had when we first started the lab, we started focusing on one of these 33 molecules called myostatin. And myostatin refers to muscle meaning myo, statin meaning stop. And so this protein was identified in the late 90s as its ability to have a negative impact on muscle. So the protein is naturally in our bodies and it suppresses muscles. And so when you knock it out or when you have mutations in myostatin, you by default get an animal with very, very large muscle mass. In the last 2 decades, there's been every major pharmaceutical company trying to identify therapeutics that could stop myostatin from working in hopes that you can boost muscle production in numerous muscle pathologies like muscle wasting diseases, muscular dystrophies, cachexia. And so what the lab really did was we focused on trying to understand the structure of this protein myostatin and how it interacts with its antagonists and also its cellular receptors. So our lab was the first lab to solve the structure of myostatin and how it interacts with some of these proteins. One of the major obstacles of targeting this family of molecules is that all 33 ligands look very similar. But as a pharmaceutical company, how do you generate a therapeutic that targets only one of them or a few of them at a time without targeting the other ones, which would then cause adverse effects if you're targeting molecules that you don't want to target. Out of the 33 ligands, we probably work about 10 to 15 different molecules in the family. And one molecule that we recently worked on called anti-Müllerian hormone, or AMH, is heavily involved in reproduction. And we just published a recent paper in PNAS on the structure of this molecule in complex with one of its receptors. And this molecule is really interesting because it plays a major role in follicle selection. And this molecule actually suppresses follicle selection in the female ovary. And it's been thought of as being used as a contraceptive, but it's also been thought of during oncofertility treatment, where women who are undergoing cancer treatment might need to suppress their follicle selection process. And so this molecule might help with that process. And then once they're done with their chemotherapy, you remove the brake on the follicle selection and allow them to have more chances for live births and allow them to reproduce after they've undergone chemotherapy. So our lab has solved the first structure of AMH in complex with its receptor.